Good afternoon. Thank you for joining us for the last of the Fall 2020 Research Compass Series events titled Data Management and Core Facilities. My name is Tiffany Willoughby, Research Education Programs and Outreach Manager in the Office of Research. I will moderate today's session. Joining me today to introduce their respective areas and share how they can offer support to you and your research activities are our guest speakers, Vanllelyn Garcia, Dr. Jimmy Gowrisanker, Cynthia Tralmer, and Bradley Woody. Here to discuss data management is Vanllelyn Garcia, a team member in the Office of Research Information Systems, also referred to as ORIS. Vanllelyn works to provide comprehensive data management services and consultation. She is happy to assist researchers with the use of Redcap, DMP tool, and ORCID, and more. Vanllelyn, welcome.
Hi, thank you. All right, well, as Tiffany mentioned, my name is Vanllelyn Garcia, and I work in the Office of Research Information Systems. Today, we're going to discuss the basics of data management. So in ORIS, we have a data management team that reviews contracts and agreements, translates any data security related regulations, and provides tools and resources for researchers to more efficiently conduct their research operations. We also have a team that creates websites for labs and centers. So be sure to check out this link I have below for a full list of resources that we offer. So today we're going to discuss what data is, what data management is and why it's so important, what a data management plan is and what a technology control plan is, and some of the research tools that we offer. A lot of times when people think of data, they think of raw data or data points or data sets, but in research, data is any information that has been collected, observed, generated, and created. So that includes physical artifacts such as models and prototypes, blueprints and diagrams, circuits, samples, software, and even audio and visual recordings. So all these different types of information are considered data. And I'm going to say the word data a lot in this presentation. So keep in mind that when I say data, I mean all these different types of information. Now, some projects or some data have certain data security standards or requirements. Most research conducted at UT Dallas is considered fundamental research. And UT Dallas BP 3096 and UT System 165 are policies that keep most of the data in fundamental research projects compliant and safe. However, there are some projects that will have HIPAA data. Currently, the Callier Center is the only covered entity that has the infrastructure in place to handle HIPAA data. You may also come across a project that has National Institutes of Standards and Technology or NIST 800-171 controls, which is quite restrictive sorts of data security requirements. And you may also encounter projects that have export control requirements. So each project is unique and has its own specific data security requirements. And that's why data management is really important. So in research, data management is the practice of organizing, storing, preserving, and sharing data that is produced in the research lifecycle. You can see here in the graph, throughout all the research stages, throughout all the lifecycle stages, storage is prevalent and important. And so that's when a data management plan comes into place. It's also called a DMP for short. So a DMP is a living document where you outline how data will be managed throughout the research lifecycle. It typically includes data description. So what type of data are you going to be producing, creating, receiving? What formats will the data be in? What tools will you use to analyze the data? Those sorts of things. And then you'll also outline if there's going to be any accompanying documentation that is required for others to read your data effectively, or metadata that will accompany your data. If you ever intend to publish any data, for example, having good metadata will allow others to find your data sets more easily. And then you will also outline how you're going to manage access to data who has access to the data, how you're going to share the data information and project materials. You'll outline if there's any ethical and intellectual property rights as part of your project. And then, of course, you will also outline how you plan to store your data, how you're going to back up your data, and how you're going to preserve your data, which also includes if you're going to preserve it for the long term or if you have to destroy it. A data management plan is very important because it improves the efficiency of research operations. If you have a plan in place, everything just will run much more smoothly. It can also be considered a sort of contingency plan. If something goes wrong, you can rest assured that you have an action plan in place that will keep data secure. So if you lose data sets, for example, you know that you have a plan that is in place to back up that data, so you won't actually lose any data. So it's those sorts of things that a data management plan will help you improve the efficiency of your operations and be a support to your research. It will also help you be more efficient in your compliance to any data security requirements as part of a project. And lastly, funding agencies are increasingly requiring the execution of a data management plan. A good example of that is the NSF. So at proposal, you're required to submit a data management plan if it's an NSF-funded award. A technology control plan is similar to a data management plan, but it differs in that a technology control plan is it is required when you do need one. So a data management plan at this time is usually highly encouraged if it's not required in a contract. But a technology control plan is a plan that is usually required when the research is more restrictive in nature. So if you have a project that's NIST 800-171, for example, if you have a project that has any export controls, you will need to create a technology control plan. And you can see it here in the bullet list. It's similar to a DMP. It does have more requirements. It's a lot more, you have to provide a lot more detail. And the data management team partners with the Export Control Office to facilitate the creation of technology control plans and review the plans along with DMPs. So here are a few research tools that we have to offer. The first one is the DMP tool. So the DMP tool is a great resource for researchers to write plans, manage plans, share the plans with collaborators so they can write on the plan together. You can request feedback on the plans using the tool. And when you request feedback, the data management team will receive a notification and will review it and offer feedback. You can keep all your plans in one place. And one thing that's great about it as well is the DMP tool has data management plan templates, a technology control plan template, and there are also funding agency specific templates that you can use as well. So it's a great resource to get yourself started and in the habit of creating data management plans. We also offer Redcap, which is a tool to collect research data. You can develop robust surveys. You can manage data sets. Fun fact is the daily health checks, if you are all familiar with the daily health checks, is powered by Redcap. So it's a very powerful tool. Okay, so we also offer the use of the Open Science Framework, which you can use to collaborate, organize, and share data with the public. So for example, NSF requires primary data and project materials to be publicly available. The Open Science Framework is a place where you can make data publicly available and share your research process, etc. We also offer Globus, which you can use to transfer large data sets securely. You can also store data in the long term, so that's a great resource there as well. And then we offer ORCID or ORCID, which you can use to create a unique persistent digital identifier, which is great because you can link it to affiliations, grants, publications, and actually the NIH now requires researchers to create an ORCID and link it to their ERA Commons account as part of having an award with the NIH. And so the ORCID is great because it allows you to pre-populate information if you link it successfully to other publications. I like to think of it as a LinkedIn profile for your research related affiliations and information. And lastly, we have a wiki page where we have documentations and tutorials. So you'll find documentation and tutorials regarding how to use some of the tools that we discussed here today. It's still in development, so be sure to check back frequently as we publish new materials. I have today. I'm happy to answer any questions. If anybody has questions, you're also welcome to email datamanagement@utdallas.edu. Yeah, thank you so much.
Thank you, Vanllelyn. There are no questions in the queue at this time. Joining us to introduce the Office of Research Core Facilities is Dr. Jimmy Gowrisanker. Dr. Gowrisanker is the Director of Research Core Facilities and therefore manages the day-to-day operations of core laboratories that support research activities across various departments on campus. With an educational background in electrical engineering and materials science, he worked in the renewable energy sector as a scientist and marketing manager. He enjoys working with students and faculty to understand their research needs, identify gaps, and provide solutions. In his free time, Jimmy enjoys watching cricket and tinkering with open source software and hardware. Dr. Gowrisanker, the virtual platform is yours.
Thank you, Tiffany. Hello, everyone. So as Tiffany mentioned, my name is Jimmy Gowrisanker. I'm the Director for Core Facilities here at UTD. Today, I want to give you an overview and a little bit about the various core facilities we have at UTD that's managed and operated by Office of Research. So the main mission of Office of Research Core Facilities is to advance the research enterprise at UT Dallas by providing researchers with state-of-the-art facilities, instrumentation, and services that expand the institution's research capabilities. So what does that really mean in terms of activities and functions? Part of the function of research core facilities is to have dedicated technical staff who are well trained to operate, maintain, and train users in the various core facilities. And the primary function is to also support translation and transformative research at UTD. As part of that, the staff also assists in method development to characterize the various samples, provide service to external academic and industrial users as well. And more importantly, the staff, we also organize relevant workshops for students, staff, and faculty. And part of the core facilities is also to identify various funding mechanisms to enhance core infrastructure capabilities. The next few slides, I will give you an overview of the various core facilities that we have. First up is the Imaging and Histology core. This is a suite of core instruments that assist in preparing samples. And then moving on to the next step is the imaging. So the imaging core we have is an Olympus Discovery Center, which I will talk about a little bit more detail in the next slide. Another core facility that's available to all users is the molecular and protein analysis, which is really an advanced, a collection of advanced optical, spectroscopic, and electron microscopy instruments that aid researchers in molecular, biomolecular, and macromolecular sciences. We also have a genome center, which is targeted towards various sequencing needs. Could be a total RNA, mRNA, or a whole genome sequencing, and targeted genome sequencing as well. To assist with a cell-based research, we have a flow cytometry core or a cell characterization core, which consists of a cell sorting instrument and a cell analysis instrument. And one of the newer cores is the mass spectrometer core, which is really geared towards identifying specific chemical species and quantifying them based on molecular weight and small molecules as well. Lastly, clean room laboratory. This is one of our oldest cores. It's a state-of-the-art class 1000, roughly 5,000 square feet, that's dedicated to fabrication and characterization of various materials, thin films, and devices for optoelectronics and MEMS devices and other electronic applications. The next several slides, I will talk about a little bit more detail, go into what specific instruments are there in each of the cores. So first up is the Genome Center. The suite of instrumentation we have are particularly with respect to the sequencing is the Illumina NextSeq, which is our next-gen sequencing platform. If a researcher is interested in single-cell sequencing, we have a Fluidigm C1. And for other type of transcriptome type of sequencing, we recently acquired a 10x Genomics. We also recently acquired Sanger sequencer. So those are kind of related to sequencing. We also have for QA and QC a fragment analyzer, Quant Studio, which is a PCR, real-time PCR, high throughput, and also Qubit NanoDrop. Services we offer in line of the library prep services, or we could provide total RNA library prep, mRNA, genomic DNA library prep, and we could provide multiple types of sequencing depending on how much data a researcher needs to collect, high output, mid output, and also provide Sanger sequencing. Yeah, a bigger part of, you'll see this as a recurring theme in all of the cores, is the training and consultation. So our core primarily provides training to users and consults with them on various sample prep techniques. If there are multiple instruments that perform similar or give similar data, our staff is here to help the users identify which instrument to use. So the next core, I'll be briefly talking about the flow cytometry core. So in this core, we have two instruments. The first one's a cell sorter. It has a plate sorting method with temperature control. The next is the analyzer, which has a high throughput plate reader. It's capable up to 128 cell, I believe. But both of these, sorting and the analyzer, come equipped with 5 lasers, as you can see, and they could, with the combination of these different lasers, you could identify 18 simultaneous markers. Similar to the genome core, our instrument training and consultation is a big part. Method development is a big part. This is somewhat of a very focused research area. So our staff here provides very detailed design of experiments. How do you design plates? How do you create positive and negative controls appropriately based on what you want to look at? And certainly the data that comes out, our staff can help with data analysis, interpretation, and we also do fee for service, which is the sorting instrument. The next slide, I'll talk to you about imaging in the histology core. So histology core and imaging core kind of go hand in hand because you really need to prepare samples first, which is what the histology provides. So we have a suite of Leica instruments, such as the tissue processor and embedder. And once you have that dehydrated and embedded with paraffin, you could, depending on what you want to image it with, if it's a optical microscope, you could choose a cryostat or a microtome that can provide you several micron thick. But if it's a electron microscope imaging that needs to be done next, you could use a ultra microtome, either at room temperature or at cryo, that can give you few tens of nanometers relevant for a TEM imaging, so to speak. We also have a suite of cell culture instruments, incubators, and a BSL2 cabinet for samples that are needed. With respect to the imaging core of the Olympus Discovery Center, this is a state-of-the-art optical imaging core. It has a very high throughput slide scanner, a confocal laser scanning microscope. If one intends to do a super high resolution, we have a spinning BIS confocal that's also capable of live cell imaging. For near cell membranes or molecular processes that need to be probed on near cell membranes, we have a cell turf. If someone wants to do intravital imaging or whole brain imaging, the optical light, due to its lack of depth of penetration, we have a 2-photon, which is equipped with an infrared laser. And it's also capable of simultaneous excitation and scanning as well. And for electron microscope, we have a Zeiss environmental SEM with variable pressure detector and EDS system for elemental analysis. And certainly, similar to the other cores, our instrument training and consultation forms a key service that we provide. Sample preparation techniques, some of the instruments or the sample preparation techniques depend on which, varies depending on which optical instrument you want to choose. So, and what instrument you select, because they all excite fluorophores and you're detecting light, but the selection of the instrument is critical depending on what kind of data you want. Certainly, we also provide assistance with data analysis and interpretation. Next, we'll talk about the molecular and protein analysis core. Like I said, this is a suite of spectroscopic and other microscopic instruments that are used in looking at chemical species. So first up, we have a Bruker 600 megahertz NMR that can handle solution and solid probe. We also have a Joule TEM microscope used for tomography and cryogenic imaging purpose. And we have a suite of Agilent FTIRs and UV-Vis used for spectroscopic measurements of various films and solids, liquids, and there are a few accessories such as multi-cell holders and temperature control that can be used in these set of FTIRs and UV-Vis. We also have, if someone's interested in relaxation studies or lifetime measurements for fluorescence and phosphorescence, we have a Horiba fluorometer that can accommodate various lasers. For any radioisotope labeling, we have a GE Typhoon for biomolecular imaging, Western blots, and other chemifluorescent measurements. And certainly for sample prep, we have several centrifuges, and we certainly have plate readers for quick identification if there's any fluorescence. Certainly, similar to other cores, we have instrument training, maintenance, and consultation. That's part of what we do here. And we also provide some guidance on sample preparation techniques. So moving on to the mass spectrometer core, we have 3 instruments. Agilent GCMS is really used for identification and quantification of small molecular metabolites. And if a sample is volatile, you can even use this. And we recently upgraded the software suite, so it has a full mass hunters for data analysis. We also have a MALDI-TOF from Shimadzu. It's really used for analysis of low and high molecular weight samples, like peptides, proteins, DNA polymers, and so on. And if you remember in the first slide, I said one of the main, one of the tasks that we often try to find is how do you upgrade or add new instruments? And we recently added a Water Xevo hybrid mass spec to this core by means of collaborating with other faculty members and users and writing a NSF MRI grant. So the fruit of that application was a addition of a new Xevo high-resolution instrument to the mass spec core that is used in studies involving metabolomics and proteomics. And this core consists a bunch of equipment and accessories to aid in sample preparation as well. Certainly, this core is more involved in sample preparation and method development, and assistance to users will be provided for such activities as well. So this is the last core that I'm going to talk about, the clean room laboratory. One slide really isn't enough to list all the instruments, so I've tried my best to kind of group them into specific function. Material deposition tools. So we have a various methods by which one can deposit metals, electron beam, sputtering, CVD, or which is chemical vapor deposition, or atomic layer deposition. Once you deposit the metals, if you want to selectively remove it, we have plasma etch tools, which are dedicated tools. We have them dedicated for metals or dielectrics. If one wants a deep silicon etch, that's possible also. And also we have a tool dedicated for 3-5, which is optoelectronics or semiconducting materials. We try to isolate them to prevent contamination. If you want to do photolithography, we have a state-of-the-art laser mask writer and a contact printer for micron-sized lithography. You can get close to micron and enough. If research calls for a sub-micron lithography, one can use an electron beam writer. And certainly it comes with various support tools like spinners, coaters, and ovens. We have a very good set of hoods for doing surface chemistry, and also a thermal processing bay. As you see in the image, optical image or photograph, an example bay looks like, and we have several bays, each focused on functionality. So we have a thermal processing bay for rapid thermal annealing or growing oxides. And the material and thin film characterization bay, we have another SEM, field emission, high resolution, with a, again, very latest EDS for elemental analysis. We have a new Oxford Nanotech AFM, ellipsometer, profilometer, microscopes, and instrument training and maintenance is our key. It's fully operated by and maintained by clean room staff. We also provide guidance on process development, but we develop standard processes and methods available for users to use, and we also use that to qualify these instruments on a regular basis. So as the last slide, you could go to research.utdallas.edu core facilities. There you could find more detailed information on various cores, the specific specifications, and what the fee schedule are for each of these cores. It also outlines how do you request training and access to any of these core facilities, how you schedule and reserve access to these instruments, and also has a list of staff members and the contact information if you should have any questions. You could feel free to email the individual core staff or me at jimmy@utdallas.edu. That concludes my presentation, and I'm happy to take any questions if you have.
Thank you, Dr. Gowrisanker. At this time, we don't have any questions in the queue.
Okay, thank you.
Next, I would like to introduce Cynthia Tralmer, Coordinator of the Institutional Animal Care and Use Committee, IACUC, at UT Dallas. Cynthia will discuss how to navigate the IACUC protocol review process and responsibilities for ensuring the humane care and use of animals in research. Cynthia has a bachelor's degree in biology from UT Dallas. She has nearly 10 years of experience working with Lab Animal Resource Center, also known as LARC, and IACUC. She formerly spent her weekends serving as a volunteer zookeeper at the Dallas Zoo and was a research assistant in a behavioral and brain sciences laboratory. Thank you for your time today, Cynthia.
Thank you, Tiffany. All right, so I will be talking to you about the IACUC today. So what is the Institutional Animal Care and Use Committee? So we are a compliance group that works for the university, and we make sure that we help the PIs remain in compliance with federal, state, and local guidelines. So who are we accountable to? We are accountable to the USDA when we house USDA animals. At the moment, we do not house USDA animals. We only house mice and rats, which are not considered covered by the USDA. We are also accountable to OLA, and if anyone receives NIH money, then we are accountable to the NIH. We use the guide as reference material for the foundation of the Animal Care and Use Program. So this really covers anything from the type of flooring we use to the type of, or the number of air changes that the mice and rats receive. So the role of the IACUC. So we review protocols, amendments and modifications, continuing reviews. So basically before you begin any kind of research with animals, or even if before you even order any animals, you need to have an IACUC approved protocol. We have the committee review it. If they have any recommendations, I will email you back and have you modify it before we send it on, and then they will approve it. We also hold semi-annual program reviews. This is part of our post-approval monitoring, and we just make sure that everything in the program is going well, and see if there's any kind of room for improvement. We also provide facility inspections. So we actually walk through the facility and ensure that everything that people are saying is going on in their protocol is actually happening, and see if there's any kind of problems with the animals, health-wise or environment-wise. We also prepare reports. So I actually just got done doing the OLA annual report and the USDA annual report. And the most important role is that we promote ethical treatment of animals used in research. So who serves on the IACUC? Well, we have an IACUC chairman, and he is primarily responsible for what happens in IACUC meetings. He actually leads the agenda, and he has a say in any kind of changes we want to make to the IACUC. We also have an attending veterinarian, and he is responsible for the health of all the animals in the facility. And he also is very useful in reviewing protocols to make sure that the chemicals used are the proper ones, and injections and all that. We also have non-affiliated members, and they are very important because I think when there is a group that does something for so long, they're kind of too close to it. And so it's good to have outside members of the public to look at the same thing and say, hey, this looks a little iffy, or what about this, and kind of bring everyone back to center to relook and evaluate things we might have looked at 1,000 times. We also have practicing scientists. So these are PIs at the institution that are currently working with animals, and they are very useful because they know what type of procedures are done in animal research and what it should look like, and they can get an idea if something is actually going to have some kind of scientific importance with the results. We also have non-scientists, so we have everything from professors that work in the humanities department to the chief of police, and they are very helpful to see the protocols and make sure that it's structured well and that it makes sense, and especially the lay summary, because it's important that everyone in the public can understand that, especially if something happens and that's released to the public. We want everyone to know exactly what type of research is going on with the animals. So how does this affect you? So as I said before, to run animal experiments or even to order animals, you must have an IACUC approved protocol. We do have facility inspections. The PIs are not super involved with this, but if the IACUC does walk through and they see a PI or a student, they may ask to see what they're doing, just to kind of follow up and see as kind of our post-approval monitoring to see if they know exactly what is going on in the surgery they're doing, and just kind of out of curiosity to see what they're doing for the day. We also ensure that all PI staff and students are trained to work with the animals. We provide BioRef training online, and we also have a hands-on vivarium orientation. And we do have surgery workshops every semester. Dr. Marcel from UT San Antonio actually comes up and will host, I think it's about 3 days now, 3 to 4 day surgery workshop for all the students. And if there's ever any kind of training you need that you're not really sure about, you can always ask me and I will find one of our technicians that can help you learn how to do a certain procedure. We also require everyone to enroll in the occupational health program. We do this by having everyone submit a medical health questionnaire and a tetanus shot. And I'll talk a little bit more about that later. And of course, building access. We require everyone to fill out a form so that we know who is in the building and who is responsible for you before we grant access to the enteral, BSB, and vivarium. So for facility inspections, we require 2 members to walk through the facility, and we're primarily looking at the health of the animals to make sure that their fur looks nice and clean and they're grooming themselves well. We look at cleanliness of labs to see if there's like corn cobs everywhere, make sure that biohazard waste is in biohazard waste marked trash cans. We also ensure that proper surgery techniques are being done if we happen to see a student or staff doing something with an animal when we walk through. And we also look at the records to make sure, like for example, chemical records, to make sure those are being done, just to give you a heads up, because the DAA will also look at those records. So it's better for us to say, hey, you need to add this before the DAA comes and takes a look. We do also have informal weekend inspections. So we have a second veterinarian, and he actually walks through on the weekend when there's less people around, just to make sure everything's going well, as there's not as much staff available. Okay, so I have posted a link to submit a protocol, and you can email those to me. We try to have a 3-week turnaround, and once the protocol is approved, I will send you an approval letter. Protocols are good for 3 years, and we do require continuing annual reviews. And this is just kind of a check-in so we can make sure that your research is on track to see if there's any new changes you need to make for the coming year. Especially this year, we've had a lot of changes with the pandemic and all that. And also to see if there's any kind of amendments that you thought you had added, but you actually had not submitted. So this information is actually going to change fairly soon. Not by a lot, but it will change. So per diems are required. It's 50 cents per cage each day for the mice and 75 cents per cage per day for the rats as of right now. And this is really just to supplement all the costs of taking care of the animals, because we purchase the racks and the food and the water and make sure that they're all taken care of. So I made this slide just for new investigators. I think it's kind of a good little step process to see how we all collaborate with each other, because it's not just me. And so if you need to get started on the protocol application, just reach out to me. I can send you that, and I can send you a breeding protocol as well if you're going to be doing any kind of breeding. And I can also let you know if we house the species of animals you want to house, and also ensure that we have the amount of space for the animals that you will be using. Also, if you need a per diem document, a lot of people ask for that, just for their grant agency to take a look at it, so they can make sure that they provide you with the amount of money to cover housing the animals you need. You can also contact Bradley, so he's actually the one that will help with the animal transfers. You can contact Tyler, so he's the one that's actually going to help you get enrolled in the occupational health program and getting building access, so he'll send you all those forms. And then if you plan to be working with any biohazards, I recommend contacting Dina, although the IACUC will usually ask when they review a protocol if they notice something. But if you want to get a head start and kind of make the process a little quicker, it's good to contact her first, and she can help you create any SOPs that you may need. And so whenever you reach out to me, I usually CC all these people, and that is why. All right, so I put my contact, and I put Caitlin McAllister's contact, and this is for reporting any kind of animal welfare concerns. If you wish to do so anonymously, there's also a link there, and that's it. Do we have any questions?
Thank you, Cynthia. There are no questions in the queue at this time. Our final presenter is Bradley Woody. Bradley will give us a more in-depth view of animal program at UTD's Lab Animal Resource Center. Bradley Woody is the Facility Research Supervisor and Animal Health Specialist who oversees the vivarium daily operations in animal health. He has worked in the biomedical research field for 8 years, starting off as an animal attendant at the University of Texas Health Science Center at Tyler. Bradley, please tell us about the Lrc.
Good afternoon. Thank you for the introduction, Tiffany. I am the supervisor here in the vivarium, and today I'm gonna give you a brief look into our UTD animal program. So our Lrc mission statement, our mission statement is to provide for the ethical care and use of animals that are utilized in biomedical research while supporting the advancement of higher education and responsible scientific progress. It is our goal to promote greater health, knowledge, and quality of life to enable individual success. Lab Animal Resource Center is a group that works in cooperation with the IACUC, federal, state, and local authorities in providing for the ethical care and use of animals utilized in biomedical research. The Lrc staff here in the vivarium consists of a facility manager, which is Stephen Lucas, of course, me, the facility supervisor, Lrc coordinator, who is Tyler Tornblum, and animal technicians. Currently, we have 5 on hand. We are all responsible for the daily care of the research animals and the daily activities here in the vivarium. Lrc group continued. We have an assistant vice president for research compliance, Lab Animal Resource Center and IACUC director, an IACUC coordinator who you met just a while ago, Cynthia, an on-call veterinarian who is Dr. Eugenie Daniels, who works his way out of UNT Health Science Center. He is also a great resource in obtaining anesthesia and for the care here at the vivarium. Vivarium operations, we're open 24 hours a day, 7 days a week to staff and students. Visitors are not allowed unless approved by our facility manager and is accompanied by lab personnel. Vivarium staff members are available 7 days a week from 8 to 5, and especially on holidays, they're available on holidays as well. For emergencies, contact campus police or the facility manager. Necessary PPE when visiting the vivarium. So Cintas provides our lab coats. Anytime that a student visits from BSB building or just coming in for the day, they're required to wear a lab coat or isolation gown, as you see on the picture on your right. We do provide shoe covers, hair bonnets, and gloves, and masks, and of course hand sanitizer for your use. In the event of emergency, we have first aid center kits in the front and back vestibule to the facility. We also have emergency shower and eye wash stations available throughout our facility, and as well as fire extinguishers. So some other emergencies, we also have like animal bites and scratches. If you got bit or scratched by an animal, please wash the affected area with warm water and soap for at least 30 seconds. First aid kits, in the vestibules has disinfectants and Band-Aids. Next, you apply to see one of us to fill out the necessary accident and unusual circumstance forms. It's basically a quick questionnaire about the facts leading up to the incident. If the wounds or bites or scratches worsens, please see the campus clinic or your doctor for follow-up. Currently, our vivarium houses 5,000 mice and rats. There's a nice picture of a nude mouse on your left. Caging and enrichment. Typical cage setup for mice and rats are IVC cages, individually ventilated cages. They're used to keep animals separate from one another and limits the potential exposures, including exposure by air, pollutants. Enrichment. Nalobones are typically used for animals' enjoyment and helps with the distress and post-op care. Nesting in red huts for mice have the same effect in relieving stress and in fact helps with the natural behavior such as nest building. The type of bedding used, the benefits of irradiated corn cob includes the reduction of allergens compared to other types of animal bedding, exhibits low ammonia levels within the cage, and the reduction of cage changes, which is great because it limits the variables, outsider variables. How do the materials get sterilized? So tunnel wash, we have a tunnel washer. It's a conveyor-like machine that has a load and unload end. Cages go through 180-degree Fahrenheit mixture of acid and cage washing compounds. The cages are loaded at the load end and they go down the line, and then at the unload end they get flipped over into the bedding dispenser in which corn cob is automatically dispensed into the cages. Aside from the tunnel wash, we have a rack washers, which is a machine that not only does cages, but washes the animal racks as well. This machine also dispenses acid wash and detergent through a series of effective sanitation cycles. We can also fit rat racks in there and rabbit racks as well. How are we sure that colonies are clean and healthy? So we have a Sentinel program, animal health monitoring program, where we take marine pathogen-free animals and place them in every single animal rack in the rooms. They are then exposed to soil bedding and feces from that colony of animals for at least 12 weeks. And then blood samples and PCR samples are taken. These Sentinel mice provide a cost-effective way to provide accurate results via blood and PCR tests, which is performed by a third party via IDEX Bioanalytics. If you ever need a copy of a health report for your colony, we can provide you the most current Sentinel results for your animals. It also is a great reference between the transferring animals in between institutions. Animal transfer requests. Questions about animal transfer requests, feel free to give me a call. This is my phone number. The link to the animal request MTA form. All animal transfer requests can be sent to me or Kathan McAllister. Adding a new lab member, Tyler Tornbloom, again, as part of our staff down here. Please, there's his email. You feel free to reach out to him in regard to health questionnaires, new person orientations, vivarium orientations, and question about animal order approvals. Facility concerns. Stephen Lucas is our facility manager. There's his email link. Feel free to reach out to Stephen Lucas in regard to animal health and facility concerns, requesting rack and housing space for your animals, and reporting animal welfare concerns. There's the three contacts of facility manager, coordinator, and myself, the supervisor. Thank you for your time. Questions?
Thank you, Bradley. There are no questions in the queue.
Thank you.
You're welcome. Vanllelyn, Dr. Gowrisanker, Cynthia, and Bradley, thank you for sharing your time and valuable information with the new faculty during today's session. Registration and so much more can be found at research.utdallas.edu. Thank you.